Welcome to my presentation on neural unbalanced optimal transport. My name is Frederike L, and this is joint work with my co-authors from ETH Zurich, University of Zurich, Microsoft Research, and Harvard University. Tracing the development of cells upon perturbation is a major challenge in biology, and the reason for that is that whenever we measure a cell, for example with RNA sequencing or microscopy imaging techniques, we typically destroy the cell. Thus, we will end up with multiple unaligned snapshots, which are samples of a cell population at different points in time, typically before and after some treatment. However, our goal is to understand the effect of treatments, such as drugs, on the level of single cells. Assuming that cells change only incrementally, recent approaches have utilized the mathematical concept of optimal transport to reconstruct these alignments. Optimal transport computes the mapping between cells that overall minimizes the cost of transporting all cells from one snapshot to the other. However, one key aspect remained unconsidered. Cells can die and proliferate as a response to treatments, which violates the assumption of conservation of mass that the classical formulation of optimal transport relies on. Therefore, this problem should be modeled as an unbalanced optimal transport problem. In this work, we present NuBot, a neural unbalanced optimal transport model, which computes a cost-minimal mapping between two unbalanced measures. Our model is based on the formalism of semi-couplings, which couple two distributions while allowing variation of mass. We propose an efficient neural optimization scheme to learn and parameterize these semi-couplings. We do this by conceptually breaking the problem into balanced and unbalanced subproblems, each tackling a separate aspect of the difference between measures. One tackles the feature transformation, so actually moving points in space, and the other one tackles the change of mass. More specifically, we seek balanced proxy measures, which are rescaled versions of the original measures, and then we want to solve a balanced optimal transport problem between these proxies. We learn these components in an alternating optimization scheme. In each step, we solve the balanced problem using the dual formulation of optimal transport, which involves two convex potentials, F and G, and thanks to a famous result from Brunier, the optimal transport maps are then given by the gradients of these potentials. As F and G have to be convex with respect to their inputs, they are parameterized by input convex neural networks. The question is, however, how do we find these balanced proxy measures? And for this, we use the current estimates of transport maps to transport a source sample to the target. And then we compare this predicted target to the actual target in order to estimate the change of mass of each point. For this, we use a discrete unbalanced optimal transport algorithm, the generalized Synchron algorithm. Our goal is to learn parameterized rescaling functions, and therefore we fit continuous versions parameterized by neural networks to these discrete estimates. This parameterization then allows us to map in an out-of-sample setting. We show the effectiveness of our method on the task of modeling the responses of melanoma cells that were exposed to various cancer drugs for 8 and 24 hours. In terms of distributional metrics, NuBot outperforms the baselines for almost all drugs. In the absence of a ground truth, and especially since we're unable to observe which cells and how many have died and proliferated, we have to rely on different approaches to analyze the predicted weights. In our data, we have two different cell lines, which are characterized by the expression of mutually exclusive protein markers, MEL-A and SOX9, and we observe very heterogeneous changes in cell counts across the cell lines and drugs. For example, for trametinib after 24 hours, we have a decrease in cell counts in the SOX9 cluster and an increase in the MEL-A cluster. Looking at UMAP projections of the cells, we can identify the MEL-A cluster and then see that we accurately predict high weights in the growing cluster and low weights in the shrinking one. Looking at all drugs together, we see a clear correlation in the sum of predicted weights per cell line and the observed change in cell counts. To summarize, in this work we have presented NuBot, a neural unbalanced optimal transport model that learns a parameterized optimal transport map between unbalanced measures by allowing mass to be rescaled during transport. Our model relies on the formalism of semi-couplings to account for variation in mass. And with this, I want to thank my co-authors and you for your interest in our work.